Implementation of tardigrade protein synthesis in 2024, a comprehensive overview. Date, March 5, 2025. In 2024, the field of bioengineering is poised for a transformative breakthrough with the implementation of tardigrade protein synthesis supported by a significant investment of $3.5 million. This initiative is designed to leverage the extraordinary resilience of tardigrade proteins for various applications across biotechnology and medicine. The implementation process is structured around 4 key steps that are aimed at maximizing the potential of these unique biological materials. 1. Research and development phase. The initial step focuses on extensive research into the properties of tardigrade proteins, which are renowned for their ability to survive extreme environmental stressors. Research teams comprised of molecular biologists, biochemists, and genetic engineers will delve into the molecular structure and functionality of these proteins. This phase aims to identify their unique characteristics such as desiccation tolerance, radiation resistance, and high-temperature survival, which could lead to innovative applications. Key objectives during this phase include characterizing the specific proteins associated with tardigrade resilience, mapping the genetic sequences responsible for their unique properties, investigating potential applications in medicine, agriculture, and environmental sustainability. 2. Bioengineering facility upgrade. To facilitate the research and application of tardigrade proteins, the $3.5 million investment will primarily focus on upgrading existing bioengineering facilities. This upgrade involves acquiring state-of-the-art equipment and technologies that will enable more efficient synthesis and manipulation of proteins. Upgrades will include advanced bioreactors for protein synthesis, high-throughput screening technologies for rapid testing of protein applications, enhanced laboratory spaces designed for interdisciplinary collaboration. These improvements are expected to enhance the capacity for research and development, significantly allowing scientists to push the boundaries of what is possible with tardigrade proteins. 3. Pilot programs and testing. With upgraded facilities, the next step involves launching pilot programs to test the efficacy of synthesized tardigrade proteins in practical applications. This phase will engage collaboration with academic institutions, industry partners, and startups specializing in biotechnology. Pilot programs will focus on developing new pharmaceuticals that utilize tardigrade proteins for improved stability and efficacy, exploring agricultural applications such as developing crops that can withstand extreme weather conditions or pests, investigating environmental applications including bioremediation strategies leveraging the resilience of tardigrade proteins to clean up pollutants. The outcomes of these pilot programs will be rigorously analyzed to assess the viability of commercial applications, paving the way for future innovations. 4. Commercialization and scale-up. The final step in the implementation process is the commercialization and scale-up of successful applications derived from the pilot programs. This phase will involve patent filing for innovative technologies developed during the research and pilot testing stages, establishing partnerships with biotech companies to facilitate the manufacturing and distribution of new products, launching marketing strategies to introduce tardigrade protein-based products to various markets. This commercialization effort not only aims to generate revenue but also to position tardigrade proteins as a revolutionary component in biotechnology with the potential to transform multiple industries. Common themes and implications. The integration of tardigrade proteins into biotechnology represents a groundbreaking approach to enhancing the resilience and functionality of biological materials. The emphasis on collaboration among researchers, institutions, and industry leaders is crucial for ensuring the successful realization of this initiative. The significance of the $3.5 million investment underscores a growing recognition of the potential economic and scientific benefits associated with bioengineering and synthetic biology. As researchers explore the vast applications of tardigrade proteins, the implications extend beyond scientific curiosity. They hold the promise of addressing critical challenges in health, food security, and environmental sustainability. Timeline of events: 2023, preliminary studies on tardigrade proteins are conducted revealing their potential applications in biotechnology. Early 2024, the $3.5 million investment is announced focusing on enhancing bioengineering facilities. Mid-2024, upgrades to bioengineering facilities commence with the installation of new equipment. Late 2024, pilot programs are initiated to evaluate the practical applications of synthesized tardigrade proteins. 2025 and beyond, successful applications are commercialized, leading to the introduction of innovative products that leverage the unique properties of tardigrade proteins. Conclusion: the comprehensive approach to implementing tardigrade protein synthesis is set to position this field at the forefront of bioengineering. By harnessing the unique resilience of tardigrade proteins, this initiative has the potential to significantly impact various fields from medicine to environmental science. The collaborative nature of the project emphasizes the importance of interdisciplinary efforts in advancing the boundaries of biotechnology, potentially leading to groundbreaking solutions for some of humanity's most pressing challenges. As the year unfolds, the world watches with anticipation for the innovations that will emerge from this exciting venture. The implementation of tardigrade protein synthesis in 2024, supported by a $3.5 million investment, reveals several counterintuitive insights. 1. Resilience versus practicality. While tardigrade proteins are celebrated for their extraordinary resilience to extreme environments, high radiation, extreme temperatures, dehydration, the practical applications of such resilience in everyday biotechnology and medicine might not be as straightforward as assumed. For instance, the ability to withstand desiccation does not automatically translate to improved performance in biological systems used in standard laboratory conditions or human health applications. 2. Cost versus benefit of investment. The $3.5 million investment aimed at upgrading bioengineering facilities might seem substantial at first glance. However, in the context of biotechnology, where large-scale projects often require funding in the tens or hundreds of millions, this investment may be viewed as relatively modest. The counterintuitive takeaway is that while the investment is aimed at harnessing cutting-edge science, the scale of funding may not align with the high expectations set for the revolutionary applications of tardigrade proteins. 3. Collaboration complexity. The emphasis on collaboration among academic institutions, industry partners, and researchers is crucial for the initiative's success. However, the assumption that collaboration will streamline the process is misleading. Interdisciplinary projects often face bureaucratic hurdles, differing priorities, and communication challenges that can slow down progress rather than expedite it. Thus, while collaboration is promoted as a pathway to success, it may inadvertently introduce complexities that hinder timely outcomes. 4. Commercialization challenges. The final step of commercialization and scale-up of successful applications assumes a linear progression from research to market. However, the reality of bringing biotechnological innovations to market is fraught with regulatory, ethical, and logistical challenges that can stall or derail the commercialization process entirely. The expectation that groundbreaking discoveries will quickly translate into market-ready products overlooks the multifaceted barriers that can impede this pathway. These counterintuitive insights highlight the complexities and potential pitfalls involved in the ambitious initiative to implement tardigrade protein synthesis in bioengineering, suggesting that while the goals may be lofty, the realities of scientific and commercial translation could present significant challenges. The report on the implementation of tardigrade protein synthesis in 2024 highlights several crucial aspects of this ambitious bioengineering initiative. While the main points are outlined clearly, there are subtler details and implications that may easily be overlooked, which can significantly affect the understanding and future trajectory of this field. Overlooked aspects and their significance. 1. Molecular structure of tardigrade proteins. The report emphasizes the necessity of understanding the molecular structure and functionality of tardigrade proteins in the research and development phase. However, the specific structural characteristics such as the presence of unique amino acid sequences or post-translational modifications could be the key to their resilience. These proteins can survive extreme dehydration, radiation, and temperature fluctuations. The significance lies in how this understanding could lead to breakthroughs in synthetic biology applications, such as developing more robust vaccines or creating crops that can withstand climate change. 2. Interdisciplinary collaboration. The importance of collaboration among researchers, institutions, and industry leaders is mentioned, yet the depth of these relationships is often underestimated. Effective collaboration can lead to knowledge transfer that catalyzes innovation. For instance, partnerships with agricultural firms could lead to the rapid application of synthesized proteins in genetically modified crops, while collaborations with pharmaceutical companies could expedite the development of new therapies. The interdisciplinary nature of this project could also enhance educational opportunities in bioengineering, fostering a new generation of scientists equipped with diverse skill sets. 3. Environmental sustainability. While the potential applications in environmental sustainability are noted, the broader implications for ecological balance and biodiversity conservation are less pronounced. Tardigrade proteins could be instrumental in bioremediation efforts, where they might help in breaking down pollutants or enhancing the resilience of ecosystems affected by climate change. The significance here is twofold. Not only could these proteins provide practical solutions to pressing environmental issues, but they could also shift the narrative around biotechnology from being merely profit-driven to being a cornerstone of sustainable development. 4. Economic and regulatory landscape. The report highlights a $3.5 million investment, indicating a financial commitment towards bioengineering. However, the economic implications of this investment, including the potential for job creation and market expansion in the biopharmaceutical sector, may be underappreciated. Additionally, as these proteins and their applications move toward commercialization, regulatory hurdles will become a critical aspect. Understanding the regulatory landscape, such as FDA approvals for new biomedical applications, will be vital for the success of this initiative, affecting timelines and costs associated with bringing products to market. 5. Potential ethical considerations. The ethical implications of manipulating biological materials, especially those derived from extremophiles like tardigrades, are often glossed over. As with any emerging technology, the potential for misuse, e.g. bioweapons, and the moral considerations surrounding synthetic lifeforms raise important questions. The significance of addressing these ethical concerns early in the process cannot be overstated. Failing to do so could lead to public backlash, regulatory pushback, or hindered research progress. 6. Long-term vision and scalability. The report outlines a timeline for the pilot programs and eventual commercialization, but lacks a long-term vision for scalability. As the field of bioengineering evolves, the ability to scale solutions effectively will determine the overall impact of tardigrade protein synthesis. This includes not just technical scalability but also the capacity to adapt to changing market demands and integrate new scientific discoveries. A comprehensive strategy for scalability can help ensure that the advancements made in the short term can contribute to sustainable long-term solutions across various sectors. 7. Cultural impact and public perception. The narrative surrounding biotechnology and genetic manipulation is often fraught with skepticism and fear. The report could benefit from acknowledging the need for public engagement and education to foster acceptance and understanding of the benefits of tardigrade protein synthesis. Engaging with community stakeholders and the public can facilitate a more favorable perception, which is crucial for the successful implementation and commercialization of biotechnological innovations. Conclusion. The implementation of tardigrade protein synthesis represents a significant step in bioengineering with vast potential applications. However, recognizing and addressing the overlooked aspects ranging from molecular specifics to the economic, ethical, and cultural implications will be crucial for the initiative's success. By considering these factors, stakeholders can better navigate the complexities of this emerging field, ultimately leading to more innovative, sustainable, and ethically responsible outcomes. 1. Research and development phase. The initial step in the implementation of tardigrade protein synthesis focuses on extensive research into the unique properties of these proteins. Tardigrades are known for their extraordinary resilience, allowing them to survive extreme environmental conditions. This phase is critical for understanding the molecular structure and functionality of tardigrade proteins, which will help identify potential applications in biotechnology and medicine. In-depth analysis of the research phase could explore the specific methodologies employed, the scientific challenges faced, and the implications of this research on future applications. 2. Bioengineering facility upgrade. The $3.5 million investment is earmarked for upgrading existing bioengineering facilities. This includes acquiring advanced equipment and technologies that facilitate efficient synthesis and manipulation of tardigrade proteins. A discussion on this point could delve into the types of technologies being adopted, the anticipated improvements in production capabilities, and how these upgrades will enable more innovative research outcomes. Additionally, the role of infrastructure in supporting cutting-edge bioengineering research can be highlighted. 3. Pilot programs and testing. After facility upgrades, pilot programs will be initiated to test the efficacy of synthesized tardigrade proteins in various real-world applications such as pharmaceuticals and agriculture. This phase will involve collaboration with academic institutions and industry partners, emphasizing the significance of interdisciplinary efforts. An in-depth analysis could examine the criteria for selecting pilot projects, the methodologies for testing, and the expected outcomes from these collaborations. The potential for commercialization and the pathways to market for successful applications could also be explored. For commercialization and scale-up, the final step involves scaling up successful applications for commercial use and establishing partnerships with biotech companies. This phase is crucial for translating research and testing into marketable products that leverage tardigrade proteins' unique properties. In-depth discussion could focus on the commercialization strategies, including patenting technologies, market analysis, and potential challenges in bringing new products to market. The broader implications of these developments for the biotechnology industry and potential impacts on various sectors, healthcare, agriculture, environmental sustainability, can also be discussed. 1. The Human Genome Project 1990–2003. This landmark project aimed to map and understand all the genes of the human species. It involved the global collaboration of scientists and institutions, culminating in the complete sequencing of the human genome. The Human Genome Project laid the groundwork for advancements in genetic research and biotechnology, similar to how tardigrade protein synthesis could revolutionize bioengineering. 2. CRISPR-Cas9 gene editing 2012–present. The development of the CRISPR-Cas9 gene editing technology represents a significant breakthrough in biotechnology, allowing for precise modifications to DNA in living organisms. This innovation has opened up new avenues for research and application in medicine, agriculture, and environmental science, paralleling the potential applications of tardigrade proteins in various fields. 3. Synthetic biology and the creation of synthetic life 2010. In 2010, scientists at the J. Craig Venter Institute created the first synthetic bacterial cell known as Synthia. This project demonstrated the potential to design and construct new organisms with tailored functions, much like the future applications of bioengineered tardigrade proteins could lead to novel solutions in medicine and environmental sustainability. In 2024, the implementation of tardigrade protein synthesis is poised to revolutionize the field of bioengineering, backed by a strategic investment of $3.5 million aimed at enhancing bioengineering facilities. This initiative will unfold in 4 pivotal steps, each leveraging the remarkable resilience of tardigrade proteins for a variety of applications in biotechnology and medicine. Future outlook and predictions. 1. Research and development R&D expansion. The emphasis on R&D will deepen our understanding of tardigrade proteins, particularly their molecular structures and functionalities. As researchers uncover the mechanisms behind their resilience, we anticipate breakthroughs that could lead to innovative applications. By late 2024, we expect to see initial findings published in leading scientific journals, sparking interest from industries seeking to integrate these proteins into their products. 2. State-of-the-art bioengineering facilities. The investment will facilitate significant upgrades to existing bioengineering laboratories, making them capable of advanced protein synthesis and manipulation. By mid-2024, we predict that these upgraded facilities will attract top-tier talent and foster collaborative projects among academic institutions and biotech companies. This could lead to a surge in patents and proprietary technologies, establishing a competitive landscape in the biotech sector. 3. Pilot programs and testing. The launch of pilot programs in late 2024 will provide critical data on the practical applications of synthesized tardigrade proteins. We foresee collaborations with pharmaceutical companies exploring drug formulations and agricultural firms testing enhanced crop resilience. By 2025, successful pilot projects could result in market-ready products addressing pressing challenges such as climate change impacts on agriculture and the need for more robust pharmaceuticals. For commercialization and scale-up, the transition from successful pilot programs to commercialization is expected to occur rapidly, with anticipated partnerships between research institutions and biotech firms. By 2025, we predict the introduction of novel products leveraging tardigrade proteins such as drought-resistant crops, advanced drug delivery systems, and materials with enhanced durability. This commercialization phase will likely attract additional investments, further accelerating innovation in the field. Driving forces behind the initiative. Economic potential. The $3.5 million investment reflects a growing recognition of the economic benefits tied to advancements in bioengineering and synthetic biology. As industries increasingly prioritize sustainability and resilience, the demand for innovative solutions will drive market growth. Interdisciplinary collaboration. Collaboration will be a cornerstone of this initiative. By fostering partnerships among researchers, academic institutions, and industry leaders, the project aims to harness diverse expertise and accelerate the development of applications that can transform various sectors. Global challenges. The applications of tardigrade proteins are particularly relevant in addressing global challenges such as food security, climate change, and health crises, as these proteins enable the development of resilient crops and effective medical treatments. Their integration into existing systems will contribute to sustainable solutions. Long-term implications. Looking beyond 2025, the advancements in tardigrade protein synthesis could lead to a paradigm shift in biotechnology. We expect that new markets will emerge as the technology matures. Entirely new markets centered around tardigrade-based products will likely emerge, including pharmaceuticals tailored for extreme environments and biodegradable materials with enhanced properties. Regulatory developments. The rapid advancement of bioengineering technologies will necessitate evolving regulatory frameworks. We predict that by 2026, regulatory bodies will be compelled to establish guidelines addressing the ethical implications and safety concerns surrounding genetically engineered organisms. Educational shifts. The growing importance of bioengineering will influence educational programs. By 2026, we foresee an increase in specialized courses and degrees focusing on synthetic biology and bioengineering, equipping the next generation with the skills needed to navigate this evolving landscape. Health and environmental resilience. Ultimately, the integration of tardigrade proteins into various applications will contribute to greater health outcomes and environmental resilience. As we develop solutions that withstand extreme conditions, we will be better equipped to face future challenges posed by climate change and emerging health threats. In conclusion, the implementation of tardigrade protein synthesis is set to usher in a new era of innovation in bioengineering. With strategic investments, a collaborative approach, and a focus on addressing global challenges, this initiative is positioned to have profound implications across multiple sectors, paving the way for sustainable and resilient solutions in the years to come.